Hello, my name is Amitesh Anand. I'm a postdoctoral researcher and I identify myself as a chemical microbiologist. I've been working in this group for last 2 years. Today I'll be talking about one of our work which has published in Molecular Biology and Evolution. This is about an innovative way by which microbes adapt to its environment and survive various stressful conditions. Life of bacteria is not easy. They have to endure various stressful conditions to thrive successfully. They often face challenge from host immune response. They always face challenges from various chemicals like antibiotic. They have to tolerate different kinds of environmental stresses like temperature, as well as their own growth stresses. It is very common for us to sense our environment and respond accordingly. It can be as simple as carrying and using an umbrella according to the weather condition. Bacteria are also capable of doing the same. They can also sense their environment and respond accordingly to endure various stressful conditions. Earlier this year, a publication from our group in Nature Microbiology showed OxyR being mutated in an iron-deficient strain when the strain was grown in iron-rich condition. After that, we observed similar mutations in two other laboratory evolution experiments of our lab. One during the evolution of one of the fastest growing bacteria, Vibrio natriegens, and other when a foreign protein is expressed in E. coli. This motivated us to find the selection pressure targeting this gene and the adaptive consequences and the physiological changes in the cell following this genetic change. It is amazing to see how microbes always find a way to survive in various conditions. We observed that all the different conditions where OxyR gene was getting targeted were having something common. Like an excess of iron causes generation of hydroxyl radicals due to the Fenton reaction. Similarly, a faster growth demand that we observed in the case of one of the fastest growing bacteria, Vibrio natriegens, so the fast growth require higher aerobic metabolic flux. Both these result in oxidative stress, and that's how there was this evolutionary selection pressure on the gene OxyR, and all the evolved strain found a way to lock this protein in a redox active conformation. Upon sensing a continued presence of a stressful environment, bacteria mutate the regulator to lock it in an active conformation. This study also provides information about the structural features of OxyR. Especially, we were able to rule out the involvement of intramolecular disulfide bond in the conformational switch of OxyR. One additional interesting information that we get from this study is what we commonly call as fear-greed trade-off. A locked-in conformation of the protein definitely helped the cells to have a better oxidative stress defense because there was no response lag. But the proteome cost due to the constitutive activity of the proteins resulted in some growth restriction. So the cells have to choose between either a greed of higher growth rate or a fear to different stressful conditions. This is very fundamental yet an applicative study. On one hand, it provides us information about mechanistic details of OxyR, information about structural features of OxyR. On the other hand, it improves our understanding of microbial adaptability. As I mentioned earlier, this study started with an observation of finding common mutation in various different projects. Seemingly, all the projects were quite different from each other. So the main challenge was to find that unifying theme between all these studies. Once we could find out that unifying feature, I had this great privilege of working with an excellent team of researchers who put together different pieces of this puzzle in a most perfect way. One of the very interesting things that we observed during the study is that the scope is not limited to the laboratory condition. We identified several natural isolates with similar OxyR mutation, which suggests that those strains will also have a much better oxidative stress ability, and that may result in antibiotic tolerant strains. So it will be very interesting to look into what kind of environments those strains have been isolated from and what are their adaptive features.